What is in an Arctic lake? Does the salt in the lake affect the life forms and how? We will be exploring these questions as part of our research with the Joint Science Education Project here in Kangerlussuaq, Greenland. We hypothesize that the salinity of lakes does affect the biology in and surrounding them. We predicted that more plankton, other animals, and plants would occur at a freshwater lake than in a saltwater lake. As part of a site investigation, we predicted that water from the saline lake would thaw more slowly due to the salt molecules hindering the movement of the water molecules.
We used the plankton net to collect the zooplankton in each lake. We calculated the amount of plankton per liter of water by determining the volume of water dragged through the plankton net of known dimensions versus the numbers of plankton caught. We used probes to determine the salinity and temperature of the lakes. We collected 2 bottles of lake water for each lake and used these samples to find pH and relative absorbance of phytoplankton.
We took measurements from 3 lakes. This here is the first freshwater lake. We saw grasses around lakeside with shrubs and moss apparent. We also found a great deal of muskox wool in the branch of the shrubs, as well as a caribou antler off to the side. This evidence shows that several species frequent the lake as a freshwater source.
This is the Saltwater Lake, at the bottom of a hill. Unlike at the freshwater lake, there were not many shrubs or grasses. We saw salt at the edges of the lake. Peat, or compacted layers of vegetation, was also abundant. In the water we found what we'll call sea kiwis, which we believe are pieces of peat rolled up by the continual movement of the water. We found evidence of birds such as the northern wheat ear, a caribou leg, and what looked like goose tracks. These signs show that the salinity of the lake did not deter animals from frequenting the area.
This is what we call sea tomato lake. It is a freshwater lake containing cyanobacteria that forms into thousands of rubbery spheres that resemble tomatoes. At the edge of the lake, the shrubs give way to rocks, grasses, and scattered flowers. There were several species of insects around the shrub. A lot of life exists here.
Of the lakes, it was time to find results. We quickly looked at phytoplankton, which in Arctic waters are microscopic organisms similar to plants that use a process similar to photosynthesis to create food. To measure phytoplankton, we used a fluorometer. A fluorometer shined an indigo light at a wavelength of 430 nanometers to excite the phytoplankton. This would cause them to shine a light green light from green pigments called chlorophyll at a wavelength of 530 nanometers. The fluorometer had a receiver that picked up these waves, and while it was not able to give us an exact number of phytoplankton, it did give us a number we could compare across the lakes. Here we can see the saline lake has the highest number with 1.162 absorbance. Next is the fresh lake with 0.041 absorbance. Finally, we have the sea tomato lake with 0.008 absorbance.
This is a breakdown of the taxonomic orders of zooplankton that we found in each lake. As you can see, calanoids, shown in purple, constitute the majority of both the freshwater and sea tomato lakes. The saline lake consisted mainly of cyclopoids, shown in light blue. The freshwater lake also contains more cladocerans than did the others, shown in light green. Also present was the ostracod, which we found in small amounts in the sea tomato lake. From the graph, we can tell that the saline lake contained fewer animals per liter, but had a greater diversity of plankton. The salt in the lake did not decrease the number of animal species inside, but caused different species to thrive in it.
Cladocera, also known as the water fleece, commonly lives in freshwater and inland aqueous habitats. We found 2 kinds of daphnias.
The calanoid is a zooplankton, which species are both marine and freshwater copepods. They are dominant plankton in many parts of the sea, making up 55 to 95% of plankton samples. They eat phytoplankton and algae.
Cyclopoids. These are small crustaceans with short antennae living in both freshwater and marine habitats. We found cyclopoids in both the saltwater lake and sea tomato lake. The cyclopod we found in the saltwater lakes is called Cyclopes rubellus and is also a marine organism. It was therefore able to thrive in this saline environment.
Ostracod, also known as sea shrimps. They have some kind of shell, and they're bean-shaped, lives in freshwater ponds.
Here you can see we found chironomid larvae, or non-biting midges. While they're not technically zooplankton, they are an important part of the sample. You can often find them in large swarms and are often mistaken for mosquitoes.
Site investigation. We froze 50 milliliter samples from the freshwater and saltwater lakes. While our results were inconclusive, it looked like the samples froze at the same rate. Here you can see the samples thawing. And in both our two trials, the samples thawed at the same rate. These results indicate that the salinity in our Arctic lake would not affect the time it takes to freeze in the winter or melt in the spring. The life forms inside it could probably undergo similar life cycles as those in the freshwater lake.
Though the freshwater lake had more zooplankton, the saltwater lake had a greater absorbance from phytoplankton. The phytoplankton in lakes is important as a carbon dioxide consumer and a huge oxygen producer. It is also a major food source for the zooplankton, which in turn contribute to the lake's ecosystem. The significance of both saline and freshwater lakes cannot be understated. We hope to impart to you the importance of these lakes, and we hope that the health of these booming ecosystems can be maintained.